All of them.
And it was like,
It was
I
just
feel like
last
they were like, it's like that's not their fault, that's your fault.
So I started writing.
Like you guys have the same materials, and it's like so you learn how those materials get ahead of you.
Especially
Because you don't know how to
it was
read.
weird.
That's what I'm thinking.
And so.
Oh, wait, scroll down to
Christy, this interview eight.
I know.
I don't know what I was
I'm so it's not as loud.
Wait,
no, but I mean, you guys have to be quiet. All right. So finishing talking today about protein structure, and then we will get into looking at biochemical techniques called spherifying protein. So when we were thinking about protein folding, the experiment and the at-home part of the lecture that we explored with the Ogdenson experiment, he explored the idea that the primary sequence of a protein is sufficient to direct protein folding. And so you could denature a protein and in an experimental setting, allow it to be refolded. And he also was exploring the disulfide bond formation, and if the disulfide bonds are required to fold the protein correctly, or if they form after the protein is already folded, which is the case. That is sufficient. So the primary sequence is sufficient to direct folding, but that is also done in the context of a test tube in a lab. When proteins are unfolded in a cell, it's a little more complicated because the cell is so chock full of protein. So if you unfold proteins in a cell, there is the potential for proteins to aggregate together, basically using the hydrophobic effect that we talked about, where hydrophobic groups like to associate because they're hiding from water. So you can imagine if you unfold a bunch of proteins in a cell, all of the hydrophobic groups from different proteins could glom together in an attempt to hide from water. So what has been shown to happen is in conditions where microorganisms are heated up a little bit, it's called heat shock, they express a class of proteins called heat shock proteins. And these are also called chaperones. They are proteins that help unfolded proteins to refold without aggregating with neighbors nearby. Why do you think they're expressed more frequently or at higher levels when a microorganism is heated up? Yeah.
Like potentially breaking the bonds, some kind of bonds within the protein that's allowing it to kind of reshape and reform.
Yeah, so when you heat up proteins, they tend to unfold because you're breaking the hydrogen bonds, you're driving that protein to become disordered. Remember the delta G of that. You're directing the protein to become, to gain entropy, that's more favorable, and you end up breaking some of the low energy non-covalent interactions that are holding together that structure. Okay, so the cells express these heat shock proteins, and we'll look at two classes of those. Their concept is the same, they're just slightly different in how they work. The first one is called heat shock protein 70, which is depicted here with no detail, just a green circle, and the fact that it uses ATP as an energy source to mediate this process. But basically what happens is if you have an unfolded protein here, the heat shock protein will bind near to the hydrophobic groups on the protein. We'll bind those. And the point there is so that you couldn't have multiples of these then aggregating together. right? Multiples of the unfolded protein. So it sort of isolates that protein by hiding those hydrophobic groups, binding to them temporarily, uses ATP energy to do that. The protein then folds up on its own correctly because it is not aggregated with others. And then the heat shock protein disengages after it's folded. Okay, so it's sort of an isolation mechanism so that the unfolded protein does not aggregate with others nearby, in which case it would probably never refold. Okay, so that is one version of heat shock protein. The other one is called heat shock protein 60, which is actually a multi-subunit protein. This is a cutaway view. So it really is a ring structure with sort of a chamber in the middle. And that's what's depicted here, sort of an open space. And this is a ring. There are two of them that sit on top of each other. So you could, the protein could actually refold two proteins in each of the chambers at one time. So the way this works is, again, sort of like an isolation chamber again. An unfolded protein would enter, and currently at this stage, the ring has exposed hydrophobic groups. So it is the hydrophobic groups inside the chamber are interacting with the hydrophobic groups that are exposed on the unfolded protein, keeping it essentially unfolded temporarily. Then we have a change in conformation. So what happens is that chamber binds a bunch of ATP and it puts a cap on the chamber so that the protein now is kept inside. And the structure changes here when that happens. When you bind the ATP and put the cap on, the internal structure of the chamber changes. And what happens there is that the hydrophobic side chains that were being exposed to the unfolded protein are sort of hid away and pulled away, and hydrophilic side chains will now be exposed to the unfolded protein. Okay, so it's changing the types of side chains that are exposed in this chamber. When hydrophilic side chains are now present next to this protein, what do you think the protein will do? What form will it adopt? Sort of like exposing unfolded protein to water. What do you think would happen? What happens to its hydrophobic side chain? They try to hide from water? Yes. So they will try to hide from water. They will fold up into the center of the protein, just like they would in an aqueous environment, and the protein will refold correctly on its own. So at this beginning stage, it's just keeping the protein unfolded until it gets in the chamber. Then the chamber changes structure, but the protein is by itself. So now it will just fold up correctly. We use ATP energy for that process, high energy phosphate. After the protein is folded, that ATP and the cap get removed, the chamber opens up and the protein leaves and the chamber resets. Okay. So sort of an isolation chamber that allows a protein to refold just according to its primary sequence, but uses the hydrophobic interactions to drive them. Okay. Questions about either of those?
So there's a step where it turns the second one.
Yeah, so this conformational change hides the hydrophobic groups that were exposed. Now hydrophilic groups are exposed, and it drives the folding. Okay, So, in a final folded protein shape, we then can stabilize its structure with different sort of final additions. These are not always required, but these are things we might see. So again, the disulfide bond formation can form between, once a protein is folded, the disulfides would form between the closest cysteine residues. Okay. You can see in the primary sequence here, they're not necessarily the ones that are next to each other in the primary sequence. They're the ones that end up being next to each other in space once the protein folds. And that was related to the Onfinson experiment. If you allowed random disulfide bonds to form before the protein folded, the protein would be incorrectly folded. Not all proteins have disulfide bonds. So this is typically proteins that are extracellular form disulfide bonds. Inside the cell, we don't get a lot of disulfide bonds because of the sulfurs are more reduced. The end groups are more reduced inside the cell. Other things we might see in a tertiary structure are metals. Ions can hold together the structure. They might be required for enzyme catalysis. You might see things like heme groups in myoglobin, hemoglobin. They're sort of big cofactors that sit in the protein. So we will see these as we go on. Lots of diseases are associated with improper protein folding in our cells. Many of these you've probably heard of. You don't need to memorize this table. One that is a really interesting, pretty devastating disease is called spongiform encephalopathy. We also probably have heard of this as mad cow disease or Creutzfeldt-Jakob disease. This is when a particular protein in our brains, which is normally expressed, has a particular structure, becomes misfolded, and it causes this very devastating degenerative brain disease. What is very interesting about this, and was not something that had ever been seen before, is that this can be caused because an abnormally folded prion protein causes a normally folded prion protein to refold incorrectly. So it almost acts like an infectious protein, right? So, so just being in the context of the unfolded protein causes this to refold and misfold. So that was something that had not been seen before this disease. So that was discovered by Stanley Prusiner, who got the Nobel Prize for that. Okay, so that is why we want our proteins folded correctly, so they don't cause disease, and so that they actually perform their functions in the cell, which they typically don't do if they're misfolded. Okay. Finally, some proteins adopt quaternary structure. In this case, this is if a protein's final form has multiple polypeptides that make up the functional protein. Not all proteins have. And so you will have terminology that would describe quaternary structure potentially. If you have two identical proteins that interact in the final product, those are called homodimers. If there are two different proteins that interact and form the functional protein, that's a heterodimer, and so on. And you can have more complex structures like hemoglobin. This has 4 subunits in its final structure, 2 alpha and 2 beta subunits. Each one has a heme group. So that's its quaternary structure and the final form. Okay, but certainly not all proteins have multiple subunits to them. Okay, any questions on the sort of wrap up of protein structure before we move on? Okay, so we are looking at some biochemical techniques for protein purification. Yes.
Excuse me. So hemoglobin has
4 identical subunits. How do we kill that?
No, they're 2 alpha and 2 beta.
Oh,
So 2 and 2, yeah.
thanks.
All right, so let's look at proteins, how we study them, where they come from, how we get access to them. So this is an example of a very complex final structure of a protein. Someone wanted to solve this structure, so they had to purify the protein and then do some structural studies on it. So where do they get proteins? We get them from cells. And this is a eukaryotic cell, but it could be from a prokaryotic cell. So oftentimes now, if people want to study a particular protein, they will clone the gene for that protein and express it at high levels, like in a bacteria. So you can make a ton of it, you can purify it, and then study it. But regardless, even if you do that, if you express it in a eukaryotic, a prokaryotic cell, you still have to purify it away from the contaminating components of the cell. Everything else in the cell, you have to get away from your protein. So we'll look at that process. Okay, so how do we extract proteins from cells? First, we have to break open the cell, and there are a bunch of different ways you could do that. 4 of them are listed here. You can sonicate cells, which is basically exposing them to high energy sound waves. All of these things are designed to break apart the plasma membrane. So you're going to end up killing the cells, and then the contents will be released. So adding detergent to break open the lipid bilayer. Brute force in one of these 2 ways. One is pushing them through an opening that is smaller than the cell diameter, or grinding them up with a mortar and pestle, which breaks open the lipid bilayer. All of those will open up the cells, and what you're left with there is called either a homogenate or a cell extract. Those are the similar terms. Okay, so it's just everything that's in the cell, but it's broken open. Okay, so now what do we do next if we have something like that? Typically, there will be a centrifugation step or 2 or 3, and we'll look at 2 different forms of centrifugation. The first one is called differential, and this is just separating the components of the cell by mass. Things that are heavier, lighter, and we can do that in a centrifuge. So you put a homogenate into a tube, you spin it in a centrifuge, and depending on how fast you spin it and how long you spin it, whatever's heaviest will pellet on the bottom, and whatever's left will be in the supernatant liquid fraction on top. So this looks at sort of 3 different steps of centrifugation. The first one is fairly low speed for 10 minutes. And in that, under those conditions, you will pellet the nucleus. Okay, so that's what is stuck to the bottom in solid form. Everything else remains in the supernatant. So if your protein is in the nucleus, fine, you can take the pellet and work further from there. Alternatively, you would take the liquid fraction here, put it into a new tube, spin higher speed, and in that way, you can pellet the mitochondrial fraction. Fine. You can take that, or you can continue on, take this supernatant and spin at very high speed. And that, at that point, you will pellet sort of these small microsomal fractions, and you would be left with all of the soluble proteins that are in the cytoplasm. So if that's where your protein is, you can take that. Okay, so that process is called differential centrifugation. Alternatively, usually wouldn't do both of these. Alternatively, you could do something called zonal centrifugation, where we're just separating the cellular components from the homogenate using density. So this is the same basic technique that we saw in the Mendelssohn-Stahl experiment, where we separated what by density. We have heavy and light. Nitrogen in DNA. Yeah, so same idea here. We have a solution, in this case, sucrose. And that experiment was cesium chloride. But basically you create a solution, you spin it and create a density gradient. So in this case, this is the gradient of sucrose. The sucrose concentration is more dense towards the bottom of the tube and less dense at the top. You create that by spinning the sucrose solution first. Yeah.
What's the method that's used to separate blood into its components?
I think so. Yeah. I mean, you can do differential for blood and just pellet the cells and then be left with plasma. So it kind of depends on what you're trying to do. If you're trying to separate just plasma from blood cells, you can do differential because that just pellets all the cells. Yeah. But you could potentially separate cellular type this way because each cell type would have a different. Okay, so if we create a gradient and then we layered our homogenate on top and then spun it again, our homogenate would migrate through the gradient and then the fractions will migrate to where they match their density up with the sucrose. So peroxisomes, mitochondria, lysosomes, et cetera, will migrate to different densities. And then you can collect fractions from the bottom and isolate different layers. So we usually pick one or the other depending on how you want to separate out your fractions, what's the best method. This one is a bit of a pain in the butt to set up because you have to create the gradient to begin with. So I would say in general, most people probably use differential because it's a little simple. Okay, great. We have partially purified our protein away from some other components in a cell, but it's definitely not purified. It's still in a big mix of other proteins. So the next step that occurs, and this is something you would do if you were in the biochemistry lab, probably. In this case, this step is called salting out. And the idea here is that if you have a soluble solution of proteins, they are different in terms of if they are more or less soluble or likely to dissolve in water. Okay? Things that impact their solubility are how many hydrophobic amino acids they have versus hydrophilic. And so each protein is going to have its own characteristics. And you can cause those proteins to come out of solution, and that's called precipitating them, by adding in a lot of salt. So sometimes a little bit of salt added to a solution of protein in water actually helps them dissolve a little bit, helps mediate an interaction with water. But at a certain point, if you add a lot of salt, you have added so much salt that it is all interacting with water. It is basically occupying all the water interaction. So the water can no longer interact with proteins, and the proteins come out of solution. They precipitate out. You can actually see that as sort of a cloudy form inside the test tube. So different proteins are going to precipitate out at different salt concentrations. So some will precipitate with less salt than others. Okay, and so you will salt out to the level which will precipitate out your protein, and then you can spin it down to the bottom of the tube. Again, you're not completely purifying your protein, but you are separating it out from some of the others. So another step in the purification process. Questions on that? Okay, and then finally, we use a process called column chromatography to do a final purification step. This is the basic concept, and we'll look at 3 different versions of column chromatography. You would usually pick one, not all of them. Okay, this one's a long lab day here. You have a column, which is essentially a glass tube, thin glass tube that is on a lab benchtop, and inside the tube are 2 fractions. One is a solid phase, and it's made of tiny, tiny gel beads. And we'll talk about the characteristics of those beads, and then a liquid phase, which is a buffer solution that runs through the tube and just drips through using gravity. Okay. You would then layer on your mixture of what was left after the salting out process. Those proteins would then move through this tube, and you would separate them out, your protein out from others. Okay, so how do we do that? How do we separate proteins in a column like this? That will depend on the characteristic of the beads and how it separates the protein. Also, important point here is, how do we know that proteins are coming off the column, right? It's all just clear solution. So how can you tell? We are measuring absorbance at 280 nanometers. Remember, that is the wavelength that and some amino acids absorb light, specifically those with conjugated double bonds. So things like phenylalanine, tyrosine, tryptophan, those absorb light at that wavelength. Okay, so if your protein has a few of those amino acids, it absorbs light. Okay, so let's take a look at the different versions of the columns we can use for this process. The first one is composed of beads that are not solid. They have channels that run through them. And what happens if you add a mixture of proteins to a column and you let it run through, this one works because the smallest proteins, meaning the smallest molecular weight proteins, will actually enter into the bead and kind of wind through almost like a maze. And it makes them migrate through the entire column slower than larger proteins because they get caught up inside all the beads. Medium-sized proteins will enter into some of the columns, or sorry, enter into some of the channels, but not all of them. So it takes them less time to go through the whole system. And then the large proteins don't enter into the beads at all. They just go around them. So they actually end up coming through first in a column. So this is called size exclusion, and it separates proteins by size, molecular weight size, how many amino acids it has. And we can measure peaks of absorbance in the fractions that have proteins coming off. Okay, so this would be largest proteins, medium, and smallest coming off the column in that order. So if you were asked, how would you separate 3 proteins? One is 10 kilodaltons, that's a measure of molecular weight. One is 30 and one is 70. You're trying to separate 3 proteins in that way. This is a good way to do that because they're very different in sizes. Okay, if you are trying to separate proteins, 2 of which are 15 kilodaltons and you want to separate them from each other, this isn't going to work. Okay? So we can use a different approach, potentially this method, still column chromatography. In this case, the beads are solid gel beads, but they have a charged group attached to them on the outside. So you can buy these beads that have a negatively charged group or a positively charged group attached to the column. And it's sort of like a fishing hook in that it will attract proteins that are the opposite charge. Okay, so if the overall charge on your protein in this case is negative, that protein will bind to the column. Okay, so this is a good way to separate proteins that have different overall charges from each other. So a mix of proteins would be added to the column here. All of the proteins that come off first would be what charges? Yeah, positive or neutral, right? Anything that doesn't bind the column is going to come off first in this big fraction. You're going to capture proteins that are negatively charged. Great. Now the problem is, if that's your protein, how do you get it off the column, right? You've captured it on the column. How do you get it? You want to temporarily change the charge on it. And so that's called eluting it. It's removing it from the column. That's what eluting means. And we're doing that by changing the pH of the buffer temporarily. If we change from a high pH buffer to a medium pH buffer, what happens to the change of the charge of the protein? What starts to happen if you lower the pH?
Or if you get protonated, like goes from negative to neutral, potentially on.
Yeah, it's the groups that can become protonated start to as we lower the pH. So the proteins that are the, I guess, least negative will come off first with less of a change of pH. And then those that are the most negative that require more protonation in order to become positively charged will come off with even lower pH. Okay, so you would then remove those proteins from the column. You want to put them back in a neutral pH soon, so you're not destroying the protein. But that's how you can remove those from the column. So that's related to the pKa's of the side chains that can be protonated or deprotonated. So that is a good approach if you want to separate proteins that have different overall charges. And then finally, our last version of the column is probably the simplest and the most specific, but you have to have a particular interaction that you know happened. This is called affinity chromatography, where a bead is modified with some sort of tag on it that binds specifically to your protein. So maybe you have an antibody to your protein, and you can attach it to the column, and that antibody will bind your protein specifically and fish it out. Or if you're looking for a DNA binding protein, you could attach DNA to the column and it will fish out a DNA binder. So you have to have some knowledge of what your protein interacts with, or you have to have a tag that's going to bind you. So it's a little specific in that way, but it's useful if you have it. You would add a mix of molecules. Everything that doesn't bind the column comes off. Your protein binds. And then same thing, in order to elute, you have to disrupt that interaction. So you might want to change the pH. You could add salt. Anything that temporarily breaks that interaction will cause your protein to come off the string. And this is an example here of an affinity column. This is a bead that has glucose attached. And it is fishing out glucose binding proteins. So anything that binds to glucose will bind the column. In this case, they actually eluted with free glucose. They just added a bunch of glucose to the column. It ends up competing for binding, and then the glucose binding protein comes off. So that's another way to elute. Okay. Questions on any of the columns there? Let's go ahead and do the problem set.